Hello, everyone. Thank you, Erica, for checking. I'm hanging on there, so we'll definitely make this session work. I really appreciate you asking, though. I hope everyone's doing well on this Wednesday evening. So, can you believe we're approaching the point where we've gotten on the good side of the semester, not too much more uh, to go, and we can say that we're actually soon looking at the halfway point of the semester. Before I'm gonna jump too much further into talking about our this week's lab, I wanted to check whether any of you had any questions about things that we might have covered um, in our last session on Monday. I do realize I was a little bit all over the place. I think that the getting unwell got the better of me. So if I didn't make any sense on that, this is a great chance to ask. I'll definitely try to clarify anything if there was anything that kind of remained a little unclear. I see a few of the students are already doing their attendance. So why don't we just make a start with that? So if you don't mind, just post your name on the chat for me, and that way I can take the attendance. I usually do it after we finish the class. I remain here writing the names down, just so that we get those updated. I also have had a chance to go through all of the coursework that you guys have submitted, and I really wanted to just say a massive thank you to all of you. I would be very surprised if you don't walk away with great grades. We've got a very driven class on this course, and I think you're gonna be positively surprised where you'll end up your final grade. So just please keep up the momentum. I know that these online classes are a little hard because there's something to do every week. And at some point, it's just easy to say that, oh, can I just have a break? But it is those little points that add up to the final grade. So just keep collecting those. And if I remember correctly, I'm just browsing at the same time on the course as I'm here. I have opened all of the learning modules at this point for you. So it doesn't mean that you have to go and work ahead of the time. But if you know that you're going to be busy, you're going to be taking a holiday or have a lot of things coming up, this should allow you a chance to work ahead of the material so that you're not going to fall behind if you need some time off. So that's the reason why I opened them up. It does not mean that you have to be stressed out. We will still keep up with the same schedule that we have kept so far. So every week, more or less, we cover one chapter and one lab. There are some exceptions of the weeks when we're doing exams. I think moving onwards, I don't try to cram so much on when you're taking an exam that week. But if you can just try to stay on top of the workload, if you fall behind, I usually accept late work as well. But really, the easiest way to not do well in an online class is if you stop submitting any stuff and then you try to cram it all in on the very last weeks or last days of the course. And I can promise that it's going to be too much to do it all at once. So just try to stay on top of the workload. Having said all that, I can't think of that I would have forgotten something, but if you do have questions, as always, shout out, unmute yourself, ask away. And what I was hoping to do today was to go through the lab that we will be completing on this week's assignments. And this lab is something that I normally love doing with my students in a classroom because it is a great chance for my students to get really hands-on involved, seeing some of the concepts that we've talked about. And I think whenever we're talking about cells, at least for me, those are kind of topics that are kind of harder to get your hands wrapped around because it's so abstract. We don't really get to see cells on daily basis with naked eye. So this lab allows us to get around that. And we actually use a real cell that's of a size that we can see. And we study how the cell membrane works using this model of an egg cell. And the model that we will be using on this lab is going to be actually a chicken egg cell. So what we end up finding is that that works as a good model of how the cell membranes of actual cells that we might be studying are selective in what is allowed to go in and what is allowed or has to stay out, and then also what's allowed to leave the cell and must stay inside the cell. So cell membrane is very selective on what it lets through and what it doesn't let through, and that's what we will be studying by using this model on this lab. So the selective permeability refer to the fact that we have either, you could imagine them as tiny, tiny, tiny holes, or the ability of the material to squeeze through that plasma membrane, but something that allows material to go in and out, while other materials are not allowed to go in and out. So we're really making, again, this membrane selective in what's allowed to move across, when, how, and in what quantities. And you might remember we revised in the last class different kinds of transport strategies that the cells might have and cell membranes might have. And one of these strategies include a situation where we had a cell membrane, which we can see here, but this cell membrane did not allow for the substance itself to travel across. What we did instead was we allowed the water to travel across this membrane. And this way, we were able to acquire a same concentration of the substance on both sides of the cell membrane. So rather than having the substance itself moving, we're moving water. And of course, if you recall back, of osmosis. So what I've done here, this is not exactly the quiz question that you'll be seeing in your lab quiz, but what I've done, I've listed the 5 most important transport strategies that we had for the membrane transport. And I would like you to feel comfortable that you can summarize each one of these in your mind. You don't have to do it in a chat, but in your mind, are you comfortable with explaining what makes each one of these different? And if you're still a little lost, I think a revision is always a good way to tackle things. So let's go through these together. And the first type of transportation that we looked at was diffusion. In diffusion, it was the substance itself that moved from area of high concentration to an area of low concentration until we had an equal concentration on both sides. So in those diagrams, it was the red dots that moved across this membrane. And of course, remember that this is an example of passive transport. This is just something that happens even if we don't spend any energy, any resources. Things just move from high concentration to low concentration until the concentrations are equal. And in this case, when we're talking about diffusion, it's the substance itself, those red dots in our diagram, that moved. The second example was osmosis, which we just saw when I introduced this lab. So again, it's going to be an example of passive transport. And as a result of that, we want to end up with the same concentration on both sides of the cell membrane. But the big thing here is that the substance doesn't actually get to move across this membrane. Rather than those red dots moving across the membrane, it's going to be water that moves. And once we have this water moving in quantities that it's the same concentration on both sides of the membranes, we're fine. That process ends. And the final example of passive transport that we discussed was facilitated transport. In facilitated transport, we had proteins that went through the entire distance of the cell membrane. So you remember cell membrane was a double layered, and these proteins extended all the way through them. So you can think of them as like little tunnels that open a direct route for materials to travel through that membrane. But this travel does not require any kind of energy investment, so it's passive, it just happens, thanks to those proteins having made a route for whatever the substance is to be able to pass through them. Of course, most proteins are selective, so they don't let all of the substances pass through, only certain ones. So that would be an example of facilitated transport. Well, then we discussed on two examples about active transport. And let's start with the pumps. So whenever we had some kind of a pump, this could be, for example, a protein, that helped to move materials against from where they would normally go. So normally what we see is that we try to have as many positives as we have negatives when we're talking about ions on both sides of the membrane, so that the overall on that side of the membrane is zero. Well, this does not always happen in human body and other animals. Sometimes we might want to establish a difference in the charges inside and outside the cell. So we actively end up pumping either negatives or positives in or out of the cell. And one example of this that we saw was that the nerve cells, neurons, which you will talk much more about on your BIO181 classes towards end of the semester, we talk about nerve cells, how nerve cells have this adaptation that they try to systematically pump ions so that they end up with a negative charge inside them. So all nerve cells have a negative charge inside them. And it is thanks to this negative charge that these nerve cells are able to then pass action potential, which are basically just electrical current, through them whenever they want to send a signal from one part of the body to another. So to establish that negative charge or positive charge on either inside or outside, that process requires some kind of pumps and active energy investment, because this wouldn't normally happen. We have to put in resources there. And the final example of active transport that we discussed was something called bulk transport. In bulk transport, we can have two different kinds of situations happening. First of all, we might have a molecule that's just so large that it's not going to be able to pass through this cell membrane. Or alternatively, it's so large that it cannot pass through a protein channel. Size of that molecule just makes it infeasible for it to pass through the cell membrane. The other option is that there's just so much of that material that we want to transport. So the volume, overall volume of the material that we want to transport is so large that we need to come up with a more clever way than the way that we have used in the past to move it across the cell membrane. And what happened? You remember that the cell membrane kind of formed a little sac on itself, and then pinched off that sac and helped to transport this material into the cell. If it was going into the cell, we talked about endocytosis. And the same process could also work by taking material inside the cell, transporting it to the cell membrane, and then releasing it to the outside the cell. That would be an example of exocytosis. So exo to exit the cell, endo to enter. And of course, both of these processes are going to be examples of active transport. We have to put in particular energy investment to make these happen. So if you feel good about all of these definitions, you're doing super well on this lab. And what I want to do next was to talk a little bit how this lab works when we do it in face-to-face classes. But before I move on to that, because I know that that's been a lot of information that we have discussed, I wanted to take a moment just to make sure to check if you have any questions that you want to ask about anything that we've covered so far. Are you all comfortable with everything that we've discussed about the cell membranes? Are you all comfortable with everything that we discussed about ways how materials can pass through the cell membranes? You don't have to be comfortable yet about how this lab works. We'll get there together in a second. And of course, like I said, this lab I think is a lot much more intriguing when you get to do it yourself in person. But because we are still kind of navigating this time with the pandemic, that some of the classes are face-to-face, some of them aren't, I've taken the best of the labs that we have, and we do a best that we can in a virtual sense out of those. But nothing prevents you. If you wanted to do this same lab at your kitchen bench at home, you could do it, and your results would be the exact same as what we end up finding on this virtual lab. So if you have any questions, just raise your hand, unmute yourself, shout out, or type to the chat. If you don't have any questions, I'm just going to move on and tell how we perform this typically in the classroom. And like I said, again, if you do want to tackle the same lab yourself, you're more than welcome to do so. It takes about I would say about a week to do the whole lab, mainly because the first step of the lab takes so much time. And I'll tell you what the first step is as we move on to this. So I told you that we're going to use a model of a cell, and this model is going to be an egg cell or egg of a chicken. Chicken lay large eggs, so it's particularly good for us. And something I really like about the chicken eggs is that they have the yolk, so you can imagine like that would be the nucleus of the egg. And then you have a rest of the white of the egg, which resembles really well the cytoplasm of the cell. So the only challenge that we're facing when we're using an egg to model a cell is that, of course, when we're using that to model an animal cells don't have this hard crust around themselves or hard shell. So we need to find a way to remove this hard shell that surrounds the egg and expose only the soft stuff inside. Well, how we do that is by using a little bit of magic of chemistry. I often tell my chemistry students that world seems very magical when you don't know basics of the chemistry, and this is one of the times that it does seem very magical until you realize that it's nothing but chemistry. So how do you remove the eggshell without really breaking the egg? Well, what you do, you will need to create a chemical reaction where you remove calcium carbonate. So the egg cell is made of calcium carbonate. And it turns out one of the best ways to remove calcium carbonate is by using acids. And one of the commonly available acids that you probably have in everyone's kitchen pantry is vinegar. When you put an egg with the shell into a glass full of vinegar, the vinegar will slowly start to break apart the calcium carbonate. As a result of that process, it also releases a gas, carbon dioxide gas, and carbon dioxide is not poisonous for us. We find that in many soft drinks and so on. So it's not really something that we would need to worry about. However, the downside of doing this, and why I did not ask you to do it at home, because I did think about it and then I was like, no one's got time for that, is that when I do this for my students in a lab, I normally prepare this ahead of time for them. And it takes anywhere between 3 to 5 days, changing the vinegar about 3 times a day. so that there's always fresh vinegar, and slowly the eggshell will get eaten away, if you wish to use that word. So that's a lot of vinegar wasted. That's a lot of time that I would put into that to do eggs and multiple eggs for each of the students. And there's always a chance that even if you do it perfectly, one of the eggs might break later on. So I did not want you to have to go through that process, but you are more than welcome to try it out at home by yourself. Well, once we have done this for anywhere between 3 to 5 days, it depends a little bit how often you change the vinegar, how thick the shell is on the eggs. There's a lot of variations on the eggs that we get. But what we end up getting is an egg that has only the membrane layer right underneath the eggshell left. So they feel really soft and squashy, a little bit like rubber balls. If you remember, those were trendy back in the day. You threw those rubber balls and they bounced back and all that sort of stuff. So now that we have done this, we have this egg that has no hard shell left. And that's what we use as a model of our cell for this experiment. And of course, what we want to study is that what's able to go in and what cannot go into the cell. So this is an actual question from your quiz for this lab. And if you have a chance, I encourage you to give your best guess of which of the following statements is not true in the chat. And this all should be based on what we just discussed. Is it true that acids can be used to dissolve calcium carbonate? Is it true that during the process of dissolving the egg cell, some gas bubbles will form? Is it true that vinegar is an example of acid? Is it true that by using vinegar, we dissolve small pores into the membrane underneath the cell? And remember that the key here was that what we did, we tried to remove the calcium carbonate eggshell. We're not trying to alter the egg itself. So the membrane underneath the shell remains the way it was. We're simply removing the layer on top of it. So based on that, of course, it would be the last option. We are not using vinegar to generate any extra pores. Those pores are there already in the egg. If any of you have ever had an egg that you've left to the fridge, for example, for a long time, or you even left it outside the fridge, eventually egg will dry up. So it will just slowly let all of the water evaporate from there through the egg shell, but also through this membrane underneath there. So if you pick that answer as not true, a really good job. And I'll always make sure to take little notes, and I'll make sure to get you those extra credits. I think that's one of the tasks that I still have on my to-do list. And unfortunately, since I got a little unwell, I didn't get around to it yet. But you will get the extra credit for active passive participation. Trust me on that. So now that we have this egg where we have removed the shell, so the calcium carbonate shell, we put this egg now in one of the 3 different kinds of conditions that we have. So every student of mine in a traditional class would get 3 eggs with no shell around them. One of them is going to be a control. So we're not going to do really anything else than put it into or keep it in a vinegar or whatever. It's not going to be something that we actually do too much about. It's there to let us see what kind of that egg was to start with. One of these eggs we place in high fructose corn syrup. So again, I think that high fructose corn syrup is a very American thing. We don't use that as largely in rest of the world, but you end up finding that in most of the pantries here, most of the stores. If you put this egg without the shell around it into high fructose corn syrup, something will happen over time. And what I do normally, I put that egg, or my students put it into a glass or a cup, and now that it doesn't have the shell, into this high fructose corn syrup. In about a couple of days, we will start seeing something happening. You can leave it into this high fructose corn syrup for even up to a week, and what's happening just will become more prevalent. But in a couple of days, you should be able to see what took place. What sort of an effect did it have if we placed that egg into high fructose corn syrup? The other treatment that we do is that we place another egg into a glass or cup of water where we have added food coloring. We add the food coloring into that cup so that we can tell if the water actually goes inside the cell or inside the egg. If the water goes inside the egg, similarly, the food coloring would travel inside the egg. So it's really there just to allow us to see that did the water travel or not. And again, we would leave this egg into this condition for a couple of days and you will already see results. Well, you probably are gonna be wondering, well, how do I know what kind of results are we gonna get? Let's have a look at that. Well, the results that we get are shown here at the picture at the bottom of the screen. The one in the middle here, that's the control egg. So it just has the shell removed, the calcium carbonate shell, it's just there. The egg on the left-hand side of the screen is the treatment 1. This egg has been soaked in high fructose corn syrup. And if you compare it to this control egg that we have, what you'll notice is that this egg is much smaller in its volume. So it has started to shrink. So that tells us that something has been moving across the membrane, one way or another. And as a result of that, the egg has lost some of its volume. And what has moved across is going to be water. So water moves to the high fructose corn syrup, trying to balance off the water concentration inside the membrane and outside the membrane. So the inside of the egg has much more water in it than outside of this egg, since outside of it is just high fructose corn syrup. And what happens as a result? Well, water travels out of the cell physically shrinks. It starts to dry up, if you wish to use that sort of language. So the overall size of this egg after the treatment 1 is going to be smaller. The volume is going to be smaller. We notice that it has given out water through its membrane. Let's have a look at the treatment 2. So the picture on the right-hand side of the screen. And here we see an egg that was placed in water with food coloring in there. And what we end up noticing is that this egg is a lot bigger than the control egg. So what we end up noticing is that now water has been traveling inside the cell, because yes, there's some amount of water inside the egg on its own, but the amount of water is not quite as much as the pure water outside the cell in the jar where we placed it. So water has been flowing inside the cell. Well, as water has been flowing inside the cell, the size of the egg has grown. Another thing that we were able to use as an evidence of the movement of the water was this food coloring that we mixed with the water where it was placed, the egg. So basically what we're seeing is that treatment 1 ends up the egg drying up, and treatment 2 ends up with the egg swelling. This sounds awfully lots of like the different tonicity levels that we discussed in our last class, doesn't it? If you did not get those tonicity levels, I will add a link to a video at the end of this recording. I enjoy those when you have a couple of good alternative recordings. That video is not from this class, it's from another class, but hopefully it will help you to kind of clarify the differences between different tonicity levels. So now, so far on this lab, we have managed to remove the egg cell. Then we put the eggs into different kinds of treatment conditions. Something has happened to those eggs, and that has allowed us to conclude that high fructose corn syrup condition caused the egg to dry up and lose volume. The treatment where we had the egg in water with some food coloring, caused the egg to swell bigger and bigger and bigger. If you have actually done this in person, you'll end up being able to actually feel these eggs in your hand. And it's kind of creepy when you realize how much that treatment 1 really dries up the egg cell. And the treatment 2, it does turn into almost like this rubber ball. Only thing is that if you squeeze it hard, now the cell membrane on the egg has been stretched as far as it can, so it can burst eventually. And this is also what would happen if we just left the egg in this condition. Eventually it would burst. So that's the lab that we do in person in class. And you've now gotten a walk through that lab. The questions from that lab are very same, regardless whether you are in person in that lab or you're taking this course in an online format. So let's have a look. One of the questions on your quiz is going to be, why do we see green food color in the egg that was treated with procedure 2? And you remember procedure 2 was this where we placed the egg into a cup of water where we had the green food color. So would it be because only the food coloring entered the egg in question? Or would it be because only the water entered the egg in question? Or would it be that no food coloring or water has entered the egg in question? Or would it be that both water and food coloring entered the egg in question? So again, if you do want to gain those points for participation, this is a good time to post your answer to the chat. What do we think that happened with that egg on a procedure 2? Was it just food coloring that entered there? Was it just water that entered there? Did no food coloring nor water enter the egg? Or was it both water and food coloring that entered the egg? And of course, good job there. It was both water and food coloring entering the egg in question. And really, I think that the last question that I had for your lab on the lab assignment that I've given to you is to consider a fact that no experiment is perfect on its own. There's always things that we can look back and say that, I would have done this a little differently. Or, I would have considered changing this. Or, I would now, based on what I know now, do this. So I'm asking in the very last question of your lab assignment for you to come up with ideas how we could improve this lab. That's not for me to see how I can do things better, but it's also very important that you know that what we show you in classes, none of it is perfect. There's always room for improvement. And I'm always wanting students to challenge what you get shown. Don't just take it as a fact. There's probably a better way to do it. And a lot of the time we have thought about those different better ways, but there's a reason why we haven't done it that way. But sometimes there's some surprising good ideas. I know that this type of a question is a new question for some of the students on 100 level biology classes, because so far traditionally you've been presented information and the answer to the question is somewhere in that information. Well, this kind of a question is now what we're expecting you to do at the college level, and once you continue to the university level, you're expected not just to find an answer in the material, but think yourself and be critical about what you're being shown. Of course, I don't encourage you to be critical in a rude way. Don't just say that it was crap, what the lab was, or that it was boring. What I'm looking for you is to find a thing that you would do differently and have some sort of a logical reason why you would do it differently. If you are really, really stuck with this question, some of the things that you can consider would be that, are all eggs alike? Do all eggs have similar size? Do all eggs have similar egg cell composition? Is it enough if we perform this experiment only once on one egg? Could we use other substances than high fructose corn syrup and water and food coloring? Things like that would be good ways to start to tackle that. How could I make this lab different? How could we find something that's a different way of tackling this lab? And that really completes everything that I have planned to share with you on this review of our lab for this session. What I'm trying to deal is with my cat, who's desperate to get your attention. But what I'm going to be doing, I'm going to be around here. If you have any questions about anything at all, I'm here to help you for the rest of the session. But I don't have anything new to share with you. So again, if you don't mind, I would appreciate if you post your name for the attendance. Again, I use those just so that I can give you the extra credits, and I promise that I will go and go through those extra credit points, and you will get them. And I'm getting a question, can I go back to the first few slides? Absolutely. Let's see. Oh my goodness, I don't know what I did there. Let's see if I get it back where I hope to be. So thank you for everyone who's posting their name for the attendance. If you don't have any questions, you can definitely just move ahead with the rest of your night. And I hope you have a very good rest of the night. For anyone who wanted to kind of loop through some of the earlier slides, I'll just go through them again, just to make sure that you get all of the information. But other than that, I'm going to wish a very good rest of the night for anyone who's off to do other things. Thank you. I really appreciate you working with me, even though I've been a little sick. Thank you. You guys take care of yourself. So in terms of the early slides, what we did, we discussed about how we can use, thank you, how we can use the egg as a model of a cell, and how egg has, similar to any other cell, it has a membrane around it. And this membrane is very selective in what it allows to come in and what it does not allow to come in. So we use that membrane around the egg as a model of a cell membrane. And in this case, we were looking at a situation where actually the substance itself didn't move, but water moved. Water where this substance was diluted in. And this was an example of osmosis. So you remember that we wanted to equalize the amount, the volume of substance on both sides of the membrane, but the substance was not able to pass through that membrane. So to equalize the concentration, we simply move the amount of water within on both sides of that membrane. The other thing that we discussed at the beginning of the session. This might have been a slide that you were interested. The other thing was that we kind of tried to have a little definition for each one of these different kinds of transportation methods, how things get transported through membranes in the body. So the first one of these was diffusion. In a diffusion, we had a substance, so the red dots in our drawings, and we had more of them in one area and less of them in another area. So we had the area of high concentration and an area of low concentration. And we wanted to make sure that the concentrations would be equal. Well, this was simply through diffusion. Over time, passively, without investment of extra energy, these move either whether there's a membrane or not, they move until the concentrations are even. That's an example of diffusion. Osmosis is a little bit of the same. However, this time we're not allowing the substance itself to cross the membrane. Rather, water moves across this membrane. So this was the example that we looked as the starting point for this lab. So the water is not moving across the membrane, only the water is moving across the membrane, but the substances are not moving across the membrane. So the only way how we can get the same concentration on both sides of the membrane is by moving water until the concentrations are even. Substances stay on their respective sides of the membrane. That was another example of passive transport. The third example of passive transport was what I described as a protein serving almost as a tunnel through a membrane. So this is passive, there's no active energy investment, but some materials are simply able to take this protein as a shortcut to go through the membrane. So this is known as a facilitated transport. So we're transporting the material across the membrane, we're facilitating that, but we are not investing energy for that to take place. Then the two types of active, I'm sorry, there's a fly or something around. Two types of active transport that we have. First one of those was pumps. So these are when we are actively using energy to generate a difference in charges on both sides of the membrane. And an example of that difference was seen in nerve cells, building a negative charge inside those cells. So sometimes using that energy is worth it because that allows that cell to do something different. And final example of active transport was when we either had a really large or a large volume of material that we wanted to transport across a cell membrane. And in this case, we had the cell membrane to kind of form a little, if you wish, a sac around the material and then pinch off from that cell membrane and carry the material inside the cell. Or alternatively, we could form that little vesicle inside the cell and then let it fuse to the cell membrane and release its substances to the outside the cell. So this would be both examples of bulk transport. And I discussed with you that we are going to use an egg as an example. However, we are going to remove the calcium carbonate shell from around the egg. And how we did that was by using acid, such as vinegar. And this chemical reaction between vinegar and calcium carbonate causes the calcium carbonate to break apart. As a byproduct, we're also producing a little bit of carbon dioxide, but that's not going to be a problem. There's just some bubbles in the liquid that you will notice. After we have done this process of replacing the vinegar around the eggs multiple times over several days, the entire calcium carbonate hard shell has disappeared. And we're just left with that membrane that's located right underneath the egg shell. And I asked a question about that, which is from your quiz, that which of the following would not be true? And the correct answer was that, of course, the use of vinegar did not do any binding to the membrane underneath the eggshell. So that membrane underneath the eggshell already had small pores in there. It's just that we're removing that hard shell from around the egg. So this statement is not true, that we would use vinegar to dissolve small pores into the membrane of the underneath the shell. That is total nonsense. We do not use vinegar for that. The three other statements, they are true. Acids are used to dissolve the calcium carbonate, and during this process we did form gas bubbles, and vinegar was an example of an acid. Then I described with you how we could use these different conditions. The control was the initial egg. Treatment 1 was when we put that egg into high fructose corn syrup, and treatment 2 was when we put it into water with food coloring in there. And as a result, we saw this outcome. We saw that the treatment 1, where the egg was in high fructose corn syrup, caused the egg to dry up. And the treatment 2, where the egg was in water with food coloring, caused the egg to swell up. So very similar to the concept of tonicity that we talked about earlier on. And one of the questions that I had, the second last question in your quiz for this lab, was that why do we see, or what do we see in this procedure 2 happening? Why do we see the green food color in the egg? Well, that was because both water and food coloring now entered into the egg in question. So what we saw now was that a movement of both water and the food coloring with the water inside the egg that was treated with procedure 2. So I hope that that helped you to go through any slides that you were kind of hoping to see again. If there's any other slides, I'm happy to go back. Just let me know. I tried to explain them, but I'm always happy to try again. Okay, I'm glad if that helped. Brittany, I really appreciate you, and I appreciate you wanting to get that. So that's I make sure to recognize that. That's all I have to share. That's all for today. I hope you have a very good rest of the night, and I hope you have a good week. Good night. And on that note, I'm gonna just stop recording. I will be around in case you have any questions later on. I will be here until about end of our normal session time. Again, I will make a recording of this session available for you afterwards. And I'm always here to help you to succeed in this class, so do not hesitate to reach out to me. I hope a good week, and I look forward to seeing you next week in our next class again. Thank you. Bye.